really excited to be here with you today to get the chance to tell you about not only what products we've developed in the last year at 10x, but the really exciting biology that our customers were enabled to do because of those products. If you're not familiar with the particular alphabet soup that follows my name, this means I'm a scientist turned genetic counselor. And so the irony is not lost on me that I will now spend the next 10 minutes or so talking to you and never say SNP, exome, or genome. And it's not because I've turned my back on disease discovery and diagnostics. That's still absolutely where my passion lies. It's just that I honestly believe that those are not the assays that are going to help us leapfrog forward in the area of treatment and diagnosis of disease. It's the kind of multidimensional data that I'm going to tell you about today that I think is really going to make a difference. So I'll start off by saying something that is quite obvious. Biology is immensely complex. But really the vastness of that complexity is difficult to even comprehend. But as biologists, we've chosen to be in a field where complexity is the norm. And for a long time, we've chosen to sort of suppress and flatten that complexity in order to make our assays easier. But I think that this has hindered us in being able to make really true novel discoveries in understanding things like, why do treatments fail? Why do some patients develop disease while others never do? And that's really what we're about at 10x Genomics, is developing the tools that help our customers address this complexity so that they can not only understand biology, but ultimately master it. And the tools that we develop, we're really committed to making tools that are easily accessible to all scientists, so that we have end-to-end solutions from our novel assays in biochemistry to the instruments that run those assays, all the way downstream to the analysis and visualization software that you need in order to make your discoveries. And we really see ourselves as being partners in those discoveries and in our customers' successes. And so we were really excited to see that so much of the really interesting biology that our customers are publishing got highlighted as the 2018 Breakthrough of the Year. Now, it wasn't just single-cell genomics that was highlighted as the Breakthrough of the Year. Most of us in this room have probably been doing single-cell genomics for quite a few years now. What science really highlighted was the novel applications of multiple assays, combinations of really interesting approaches that enabled us to understand biology at a level of detail that we had never been able to before. And as science highlighted, the single-cell revolution is just starting. What I'll talk to you about today, the kind of biology that our products enable, are really just the tip of the iceberg in terms of what we can discover with these products with single-cell genomics. These are just a few of the publications that our customers were able to put out in the last year, combining our products to make novel discoveries. And I'll highlight a few of these in more detail today. We're excited to say that we just had our 10 millionth cell published, although there's at least as many cells that we know who are making their way through the publication process. So I'll be really excited to see how many cells we have published by next year. We like to do things exponentially, so let's go for at least 100 million. And because we really see ourselves as partners in your discoveries, we like to deliver on what we say that we're going to do. So last year we told you we had a pretty ambitious R&D plan to release multiple new capabilities and assays. And I'm happy to say that we did that by releasing 6 new major capabilities on our platform, including a single-cell copy number DNA platform, a chromatin accessibility single-cell assay, the V3 version of our single-cell gene expression assay, as well as a feature that we call feature barcoding that Ben alluded to, which allows you to barcode any feature that you may be interested in reading concurrently with gene expression or immune profiling information. And again, because we're committed to the kind of end-to-end solutions that make these assays easy for everyone, we've partnered with multiple other companies to make reagents that are available off the shelf to do things like detect cell surface proteins, to detect antigen specificity, and CRISPR guide RNAs. And coming up, Mike and Tarjay will tell you about some really exciting developments that have come out of 10x in the last few months and in the last year, including a truly revolutionary data set that we'll be releasing soon that Mike will tell you about. So I want to highlight the kinds of biology that single-cell genomics really enables. And I'm going to start with one that I think we often think about as sort of the lowest common denominator of single-cell genomics, or identifying cell types in a population. And I think we too often think about identifying cell types as a necessary first step toward doing something else. But in fact, I think this is a really beautiful example where profiling cell types can lead to discoveries that are immediately useful to the disease community. So in this study, researchers from Aviv Regev's lab were looking at the mouse trachea for what kind of cell types were present. And what they found was a very, very rare population of cells that they called ionocytes, present in less than 1% of the entire lung. So this is a population of cells where you really need the kind of scale that the 10x solutions provide if you're going to see a population at less than 1% frequency. But what's truly exciting about this cell type is that it expresses more than half of the lung's CFTR protein. So if you've followed cystic fibrosis at all in the last few years, it's often the case that people focus on the ciliated cells of the lungs as the targets for things like gene therapy. But in fact, those are not the cells that express the majority of the CFTR. And so now we have this new cell population that becomes immediately relevant to disease researchers and may in fact provide novel treatment options for these patients. But oftentimes, a single assay can't give you the entirety of the information that you're interested in. In this internal data, we took samples of peripheral blood mononuclear cells and split them to perform two different 10x assays. The single-cell gene expression assay, as I just showed, and our single-cell chromatin profiling solution. And so what we see, and what we were really asking here, is if a transcription factor is expressed by a cell, in the simplest case, you might think that the motifs for that transcription factor would then be open in that cell, and that's how the regulation would proceed. Of course, as I started off by saying, biology is so much more complex than that, and so we see lots of different regulatory patterns that provide a sort of elegant complexity to this entire system. So what we show here on the left is a restricted pattern of motif accessibility. Whereas on the right, you can see that this ELF1 transcription factor is in fact expressed pretty ubiquitously, and so the regulation is really occurring more at the level of motif accessibility. We also see cases of the opposite, where the motif is quite open or red in the majority of the cells that were profiled, whereas the transcription factor expression itself is quite limited to smaller cellular populations. In another really nice example of how combining information from multiple assays gets you additional information, this is a bioRxiv paper that was published by Hanli Ji's group, in which they did single-cell DNA sequencing to identify copy number variation, as well as single-cell RNA sequencing on the same primary tumor, in this case, a gastric tumor. And what they were asking was, what does the interplay look like between the CNV profile of a set of cells and their expression profile? So what I've sort of drawn a little box around here are the cells that have copy number variation. So this is your presumptive tumor in your sample, and you can see two little blue areas below that, which are the diploid or probably normal cellular populations. And you can then ask, what is the kind of expression differences between these clusters of cells? We can see that EpCAM is expressed only in the tumor population. And then we can ask, well, what are the other infiltrating normal cells in this sample? And you can use markers like THY1 to find your myofibroblasts and PTPRC to find the immune cells that are present in this sample as well. So now moving beyond just combining assays together, this is a really interesting publication that came from Jan Jonker's group, in which they used a clever little trick with Cas9 to look at developmental tracing in zebrafish embryos. And so what they did was they injected Cas9 with a guide RNA for RFP in a zebrafish line in which there are multiple random RFP integrations. What they didn't provide was the homologous repair template for Cas9 to use. And so what happens in this context is Cas9 induces little insertions and deletions at the guide site, so within RFP. And these can now act as heritable barcodes. But because Cas9 is actually active for multiple hours over the development of the zebrafish embryo, you get barcodes upon barcodes. And so by using concurrent single-cell gene expression information, you can start to imply what cells are derived from each other and develop these sort of graphics of the lineage trajectories of cells in the early zebrafish embryo. And finally, I want to end with an example from Aude Chapuis's lab, in which they were looking at mechanisms for resistance to immunotherapy. They were using melanoma in this particular publication. And in this case, what they were doing was injecting T cells that had been engineered to recognize an oncoprotein that drives this particular type of melanoma. Unfortunately, with immunotherapy, too often patients initially respond to treatment and then eventually acquire resistance to that treatment. Most of the time, we don't know why patients develop resistance, and it makes it really hard to choose what your next step therapeutic will be. But in the few instances in which we have understood why patients develop resistance to immunotherapy, it's been because the injected T cells are no longer present, and so they can no longer attack the tumor. But because in this publication they looked at serial gene expression and immune profiling over time in the patient, they could see in the cluster indicated by the arrow colored in red that the T cell population was quite prevalent during the response phase of the tumor, but it was still present during the resistance phase as well. It's not quite as prevalent, but it's still there. And so we have to ask, well, then what's causing resistance? They did DNA sequencing of the tumor post-treatment to see if they had induced any new somatic mutations that might explain the resistance, and there was nothing there. What gave them the answer was single-cell gene expression. What they noticed first is that the transcriptional profile of the tumor pre- and post-treatment were vastly different, such that they cluster apart from each other completely. And one of the genes that drives that difference in expression between the two clusters is HLA-B. So the tumor had transcriptionally suppressed HLA-B. There was no DNA mutation to explain this behavior. The tumor had shut off HLA-B, but not HLA-A, which means that standard immunochemistry procedures were not going to be able to identify this loss of HLA-B. But what's really exciting from a clinical perspective is that this transcriptional suppression is reversible. And so this provides us a new line for which we can pursue to gain insights on how we might be able to handle resistance in patients who have become resistant to immunotherapy via this mechanism. And they found at least one other patient in which the same thing had occurred. So what's next? I think that's where we have to ask you the question. What information would make your next discovery possible? If it were me, I would guess it's going to be some combination of doing multiple assays in the same cell or in the same sample, and then ultimately remembering that cells don't exist as these dissociated blobs, that they actually have a spatial context, and that understanding that context will be critical in understanding how diseases develop and what treatments we can use to push them forward.